Hey guys, so today we're going to start chapter 10, and that's all about DNA replication. We're going to go over sections 10.1 and 10.2 in this lecture video. So, DNA replication. So now that everyone figured out, like in 1944, they figured out that DNA was a genetic material. 1953, they figured out the structure of the DNA. Now they actually wanted to see how the DNA carried out its function, right? And one of the main things that genetic material needs to do in order for genetic continuity is for DNA to replicate or duplicate, make itself into two clones. So they wanted to really figure that out. Now, Watson and Crick had already provided the basis of this because they had already stated and showed how there was complementary bases between the DNA strands. And so the DNA knew that if they found an adenine, a thymine would have to stick next to it because those two were complementary bases. So they already provided pretty much the fundamentals to it, but there were still a couple of things to clarify. Namely, if the DNA replicated semi-conservatively, conservatively, or dispersively, which we'll get into in just a second. So I'm going to share my screen right now. If I go, yeah, here we go. I'm going to share my screen. Is this what you can see? Sorry, hold on. Yep, that's what you can see. So here we go. So this replication of DNA is an essential function of genetic material. It has to be a process that is incredibly efficient, right? If you don't want to make any mistakes within the DNA, that means that you have a mistake within the whole entire organism. It's a severe mutation. And so amazingly enough, even though our DNA replicates, the DNA replication is such an immense feat, especially in any kind of organism, but even if you compare it to us, right, just to, like, if you think about human genetics, one of our cells has about 3 billion base pairs. And so that is insane. Every time that cell has to divide and go through cellular division, the DNA must replicate all those 3 billion bases or base pairs. And so it's a very humongous feat. And even though it's such a huge feat, there really isn't that many errors that are made. There's actually very minimal amounts of errors. The errors are about 1 every million base pairs. So that's equivalent to about 3,000 mutations every time the DNA replicates, which really isn't a lot, again, considering the immensity and how big our actual genome is. So it's a very efficient process, and we'll see why it's efficient. The various enzymes that are necessary for DNA replication, specifically the DNA polymerases, do have like this proofreading quality about them. So they basically go back and see if there's any errors that have been made. And it's just a very quite complicated process. It's a very complicated process, but it's made simple in its own way, I guess you could say. So the three models that we had were the conservative, like that they had before they figured out and before they understood how replication worked. Everything had a beginning, right? So the three models that people went on were the conservative, the semi-conservative, and the dispersive models. Watson and Crick, remember those guys who basically figured out the structure of the DNA, they had proposed like literally a month later, so in May, they proposed a month later that they thought that DNA replication happened semi-conservatively, just because it made sense in their mind. In a semi-conservative model, each replicated DNA molecule consists of one old and one new strand. In a conservative model, the two newly synthesized strands come together and the original helix is conserved. And then dispersive, this is the one that really didn't have a lot of support at all, just because it requires so much energy and is just very inefficient. But with the dispersive model, the parental strands are dispersed into two new double helices. So I'm going to change camera right here because I already drew it out. So with the semi-conservative, if this is my original, so my original old strands, right? They would unwind, separate, and then each strand would act as a template for the enzymes necessary for DNA replication to read and basically add the correct complementary base. And so we'd have A here, the old strand, and then the new strand being made, and I've made it in a different color in purple, the new strand would be made. And this is where the semi-conservative model basically stops, so that each replicated DNA molecule, see we have 1 and 2 here, okay, would be half old, half new. Okay, half old, half new. So this is semi-conservative, half old, half new. Conservative would mean that you'd go through the same process, and then at the end here, after our newly synthesized DNA strands were made, the new would pair up with basically disattach and pair up with the new. Then one new strand will pair up with the other new strand, so like this. And then the old would pair up with the other old strand. So like so. So we'd have one DNA, I mean, they're both identical, but one of them would have two new strands and then the other one would have two old strands. The dispersive is a mess. Basically, you'd have some parental or old bases and some new bases just kind of dispersed everywhere. We'd start off with something like this and then it would all jumble up. And as you can imagine, this jumbling, this craziness, probably something that would not occur. And in fact, I'll tell you right off the bat, dispersive doesn't happen, conservative doesn't happen. What happens with DNA replication is a semi-conservative. So Watson and Crick were correct, but as per usual with anything when it comes to biology, you need to make sure that you have evidence to provide your hypothesis, like to back up your hypothesis, right? So this is when we had the Messelson and Stahl experiment. These guys it happened in 1958, and they experimented on E. coli cells, so some bacterial cells, and they were able to actually prove that DNA replication happened with the semi-conservative model. How did they actually do that? I'm going to stop sharing again, but we are on slide 4, right? Slide 4, 5, and 6, actually. So how did they actually do that? They did that because they had two types of nitrogen. Okay, there's two types of nitrogen in this scenario. Okay, we have the 14N, which is pretty much your normal nitrogen, okay, which has original density or a specific amount of weight to it. And then we have the 15N. This is a nitrogen isotope. It has an extra neutron. Neutrons are heavy, and so this means that the 15N is heavy with respect to the 14N. So this is lightweight, so less heavy. Okay? So 15N and then the 14N. And what they ended up doing was they purposely grew the bacteria, the E. coli, in a medium that provided the only nitrogen source was provided by ammonium chloride. So, oh my goodness, I don't know if I'm going to get this correct. Ammonium chloride, which is something like this. Is it NH3, NH4? I don't know. Yeah, ammonium chloride. Sorry, it's NH4. I just looked it up. NH4, and then the nitrogen was specifically the 15N isotope, so the heavy isotope. And so this means that the bacteria, it needs nitrogen, and all the molecules that require nitrogen are going to basically take up and include this heavy nitrogen isotope. Okay, so any kind of molecule, especially molecules like DNA, right, which have nitrogenous bases. So all of the bacteria that grow on these petri dishes, if we kind of make a zoomed in picture of them, in their DNA, right, they all have, their DNA is 15N. So their DNA has a heavier isotope of the nitrogen. Okay, so this is my E. coli. All right, so this is it. They then took this bacteria and they grew it in a medium, always with ammonium chloride, which I now know is like so. That's the molecular formula. But here the nitrogen was no longer the heavy kind. It was the less heavy kind, normal isotope. Okay, it was a less heavy kind. And so when the bacteria would reproduce, the bacteria originally had the 15N. It would slowly incorporate inside its DNA the 14N, right? So, and then they were able to basically see they could distinguish the 15N DNA, distinguish it from the 14N DNA with a technique known as sedimentation equilibrium centrifugation, where basically they centrifuged the DNA, and the DNA that was heavy, and it provided this specific kind of density gradient, and on this density gradient, the molecules would stop at where they'd find, like where their own density would be in equilibrium with that density gradient. I don't really care that you know too much how the sedimentation equilibrium centrifugation worked, but you do need to know that after centrifugation, any DNA that had the N15, right, the 15, and I always write it at the end, it's actually at the beginning right here, so I apologize for that, with sediment to the bottom because it was the heaviest, okay, it was the heaviest, and then afterwards, the DNA with the 14N was sediment a little bit less because it was less, like a little bit above the N15 because it was less heavy. Okay, so now we are growing bacteria that have the purple 15N, they're heavy. Okay, these bacteria are going to start duplicating. And when they duplicate, they have to duplicate their own DNA. And so they're going to incorporate the only nitrogen source that they have, which is going to be that less heavy 14N. Okay, so if we go back to the experiment, share the screen. If we go back to the experiment, it was just an experiment that provided strong evidence to the fact that we had actual conservation, semi-conservation, sorry. So we had our E. coli that had the 15N in the nitrogenous bases, okay? And we added it to the 14N medium, okay? And we allowed the bacteria to actually replicate. Oops. Yeah. And so when the bacteria replicated, right? If this is our original 15N DNA, when the bacteria replicated, if it was a semi-conservative model, which it is, one of the strands would have the 15N and the other strand would have the 14N for both of the duplicated DNAs. Can you guys see that? Okay, so one strand had the 15N, the old, and then the other strand had the 14N. So when you take one of these molecules, they have basically an equal amount of the heavy isotope and the less heavy isotope. So they're going to give us like an intermediate molecule. And the same can be said about here. So when we went through, when they underwent centrifugation, what they saw is that they got a band kind of like an intermediate band. So it would be, if you guys can see right here, the band would show up somewhere in the middle. So between the 14N and the 15N. So somewhere in the middle, not at the bottom, which is where we have this one, right? Not at the top, which is where we have the 14N. Somewhere in the middle, because we had half of 14N and half of 15N. I should have made 14 green, sorry. There we go. Half of 14N and half of 15N. And then they saw that if they continued, so the second generation, if they continued to allow the E. coli to grow in that 14N media, with the second generation, now they actually had more bands come up. Some bands had only, the DNA was only 14N. So if you think about this one DNA molecule separating, so you have the old 14N strand and the old 15N strand, we still have an old 14N and then a new 14N. So now we only have one molecule with only nitrogen that is the 14N isotope. And then here with that old 15N, we now have another 14N. So this is how we got two bands, right? We got 14N, 14N band, and then the intermediate band right here. So this basically proved that it was semi-conservation. If it wasn't, if it was supposed to be something like the conservative model, you'd see that with the conservative model in that first generation, that first replication, one of the DNA molecules was only going to be 15N. So it'd have one 15N strand and another 15N strand. And the other new one was going to have one 14N strand and another 14N strand. That did not happen because in the first generation, that first replication cycle, we got a mixture between 15N and 14N. So the conservative model was kicked out. Still at this point with generation 1, we couldn't actually negate the dispersive replication model. But when we looked at generation 2, we could start seeing kind of that it was definitely only the semi-conservative. Because if it was the dispersive model in the second replication, we wouldn't have any DNA that would solely be only 14N. You see that? We wouldn't have any double-stranded DNA that would solely be 14N. It would always be kind of a mixture between the parent and the original. So if the replication was dispersive, all generations would mean that the DNA would have to have an intermediate density. So we would only show up with this band right here, this purple band, and this blue band would never show up, right? It should basically be that way, that we'd only have this intermediate band happening. However, that was not the case. They saw that there was DNA that had two strands of 14N, two strands, one of the with each having 14N nitrogen, and therefore they could basically they sufficiently disproved this experiment with like this, not the experiment, they disproved the dispersive model. So all in all, the Michelson-style experiment basically proved that it was due to semi-conservation, right? It was due to semi-conservation. Again, though, this was only something that had been seen in E. coli, in bacteria, and so we needed something to show that the same occurred in eukaryotes. And this is when Taylor, Woods, and Hughes came about in 1957. They basically conducted an experiment on broadbean, I don't know, a bean. I'm really not sure what broadbean is, but a bean plant. Okay, they conducted the experiment on the root tip of a bean plant, and they showed that the DNA replication here was also semi-conservative. They use a very similar kind of experiment, except that this time the DNA was tagged through autoradiography. They grew the DNA in a specific, so that it was labeled with this, this 3H thymidine, which was basically a radioisotope, right? And so with this radioisotope, they could detect it from all the other normal non-tagged DNA. So in the experiment, they grew the root tips in the radioisotope presence. Okay, so they grew the root tips, the unlabeled chromosome. They grew the root tips in the radioisotope presence, and then they placed. So now that the DNA it was all radioisotope basically, and then they placed this DNA in an unlabeled medium where cell division was continued. At the end of each generation, they added this drug known as colchicine. We actually talked about colchicine at the beginning of the genetics lab, right? Colchicine stops basically mitosis from happening. It arrests the cultures and actually arrests them at the stage of metaphase. And so they added colchicine to stop the DNA DNA replication had happened, but they stopped the cell division from proceeding. And so they were able to basically analyze and examine the chromosomes by autoradiography. Now, autoradiography helps pinpoint the locations of the radioisotope in the cell, so they could find where this radioisotope, that 3H thymidine, was, and they can make photographic emulsions placed over the cellular material, and then it could be developed into a photographic film, and they could basically see the radioisotope would emit energy and show up as darker spots compared to places like DNA that didn't have any radioisotope in them. And so essentially what they saw was that they found the radioactive thymidine. It was only associated with chromatids that had newly synthesized DNA. Okay, so as you can see here, right, half of the chromatid had the radioisotope, the other half didn't have the radioisotope. After the first generation, both sister chromatids showed radioactivity, indicating that each strand contains one new radioactive DNA strand and one unlabeled. And then after the second generation, which you can't see here, well, yeah, this is the second generation right here. After the second generation, which occurred in the unlabeled medium, only one of the two sister chromatids of each chromosome should be radioactive or is radioactive, because half of the parent strands are unlabeled. Okay, so it's pretty much the same concept as the Michelson and Stahl experiment, except this time they used radioisotopes, and they used it in eukaryotes, specifically this bean plant, right? And so again, it was compatible with the semi-conservative replication, and similar technique to Michelson and Stahl, had the radioisotope labeled root tip grow in an unlabeled medium, and the results showed that in the first generation, the radioactive thymidine chromatids were in association with unlabeled chromatids, so what we had here, right? And again, compatible with semi-conservative replication, and later studies continue to have reached the same conclusion. So they were all in great support of Watson and Crick's original, I'm going to change video, I didn't realize I didn't change video, sorry. They were all in support of Watson and Crick's original hypothesis, essentially. So DNA, semi-conservative. Okay, so that was basically the end of the experiment that told us that our DNA replicates in a semi-conservative manner. Then the other questions they started looking into is, like, does DNA replication start at one point of a DNA molecule, or does it start in multiple positions? How does it go? Does it go in one single direction? Is it bidirectional? Does it go in two directions? So they basically carried out more experiments to try and figure this out. essentially though, replication does begin at particular sites, especially like in terms of bacteria. These sites are called origins of replication, or ORIs, right? So there's these short specific DNA segments on the DNA, and this is where the replication commences. And when it does commence, the DNA, so the DNA is this whole long thing, right? So this is my double helix, whole long thing. And so they say here the watch is where my segment of DNA, the origin of replication, that segment of DNA where replication actually starts. Here, this is going to start unwinding. Oh my gosh, I'm doing a terrible job. This is going to start unwinding. And so now we can start, the two strands are there to act as templates for the newly synthesized DNA. And so when the DNA unwinds, you can see it a lot better here. When the DNA unwinds, you start forming this, what we call a replication bubble. And at this replication bubble, we have two forks, okay? They create two forks at each end of the replication bubble. And this is just a Y-shaped region where new DNA strands are elongating. And so as the DNA strands elongate, this fork just basically keeps getting like growing, getting deeper and deeper into the DNA. So DNA replication begins at the origin of replication, and it is also bidirectional rather than unidirectional. So when our DNA, right, when it opens up and DNA replication starts, it doesn't just go one direction, it goes, the DNA is replicated in both directions. So you can see here we have two replication forks. Each of the replication forks is where the DNA is going to elongate. So we have one elongation happening on this side, as you can see by the red arrow, and then there should be another arrow, be nice. There should be another arrow on the other side indicating that DNA replication happens on this side as well. So it is bidirectional rather than unidirectional. Two directions, not just one. So replication proceeds in both directions from the origin. So replication forks are migrating. They go further and further apart from each other. They basically go deeper and deeper inside the DNA that needs to be replicated. And then once the entire molecule of DNA is replicated, or that's known as the replicon. The replicon, this is just a length of DNA that is replicated. So if we see here, bacteria have one ORI. We've talked about this already. Bacteria have circular chromosomes. So if they have one ORI, you open this up, as you can see, you open it up, you have two replication forks because of that bidirectional replication, and it just proceeds, one replication proceeds one way, so to the right, and the other replication proceeds the other way to the left, until we replicate our DNA all the way until we get to that end point. That end point, that termination region, is known as a TER. It's called the TER region, okay? And then basically the replicon, remember, is that DNA, the replicated DNA, it consists of the entire genome, which in E. coli is about 4.6 million base pairs. And so therefore the entire bacterial chromosome constitutes one replicon, essentially. So we have 23 pairs of chromosomes, so 46 chromosomes. We technically have 46 replicons. So it's the 46 DNA molecules or chromosomes that are going to be replicated. And so with eukaryotes, depending on the number of chromosomes we can have many, many more replicons. Usually bacteria have one single replicon, which is just their entire genome, right? So this is actually a really good thing to think about, the difference between prokaryotic cellular division or bacteria. This, should not be capitalized. There we go. That bothers. So the replication of bacterial DNA and the replication of eukaryotic DNA. So bacterial DNA is a circular chromosome. So they have one origin of replication, and then with that when that replication bubble is made, the replication forks migrate all the way down until all the DNA is synthesized till they get to that TER, that termination site, right? And then we get two daughter DNA molecules. With eukaryotic cells, our DNA is linear. It's not circular, it's linear. And it's a lot longer usually. And so we have multiple origins of replication. So multiple replication bubbles open up, and they basically kind of merge into each other, just kind of aiding each other and helping the DNA replication process go by faster until we have two daughter DNA molecules. So I'm going to end right here. And then, so I lied at the beginning. We're only doing section 10.1 right now. And I'm going to talk about section 10.2 in the next lecture.